Welcome back to G Fun Facts Online. Today we are diving into the microscopic architecture of your brain to uncover a story about geometry, aging, and a molecule with a name that might make you giggle, but whose function is absolutely critical for your survival. We are talking about the Spermine Defense. In the labyrinthine biological architecture of the human brain, a silent war is constantly being waged. It is a battle of geometry. A struggle to keep the billions of proteins that power our thoughts and memories folded in their correct, functional shapes. When this folding process fails, the consequences are catastrophic. Proteins that should be neat, functional machines instead collapse into sticky, toxic tangles. These are long, spaghetti-like fibrils that choke neurons and lead to the devastating cognitive decline seen in Alzheimer's, Parkinson's, and other neurodegenerative diseases. For decades, scientists have searched for a way to stop this process, which we might call the spaghettification of brain proteins. They have looked for drugs that can cut the fibers, or antibodies that can flag them for destruction. But a recent breakthrough from the Paul Scherrer Institute in Switzerland suggests that the solution might have been hiding inside our cells all along. It involves a tiny, ancient molecule called spermine. This molecule acts as a powerful defense mechanism. If toxic brain proteins are the unruly spaghetti, spermine is the cheese that binds them together, not into a sticky mess, but into manageable, tasty morsels that the cell can easily digest. This is the story of the Spermine Defense, a molecular mechanism that challenges our understanding of aging, protein folding, and the very nature of how our brains protect themselves. To understand the defense, we must first understand the defender. Spermine is a polyamine, which is a class of small organic compounds having two or more primary amino groups. Chemically, it is simple, just a short carbon chain peppered with nitrogen atoms. But biologically, it is profound. Polyamines, like spermine, are ubiquitous. They are found in almost every living organism, from the simplest bacteria to the most complex mammals. This evolutionary conservation suggests they play a role so fundamental that life itself can barely exist without them. Spermine was actually first discovered way back in 1678 by Antony van Leeuwenhoek, the father of microbiology. He observed crystals forming in human semen, hence the name spermine. However, it took nearly 250 years for its chemical structure to be fully elucidated. For a long time, it was viewed merely as a metabolic byproduct or a simple growth factor, but we now know it is much more. Your body manufactures spermine through a tightly regulated assembly line. It starts with the amino acid arginine, which is converted into ornithine. From there, the enzyme ornithine decarboxylase creates putrescine. Putrescine is the base. Then, another enzyme adds a molecular group to create spermidine, a crucial molecule for longevity that you might have heard of. Finally, spermine synthase adds another group to create spermine. This hierarchy is important because these molecules can interconvert. Your cells constantly balance the levels of putrescine, spermidine, and spermine. If one is missing, the whole system can crash. The secret weapon of spermine is its positive charge. In the neutral pH environment of a cell, spermine carries 4 positive charges. Most large biological molecules, like DNA, RNA, and many proteins, are negatively charged. In the world of molecular physics, opposites attract. Spermine acts as a portable cationic shield. It zips around the cell, sticking to negatively charged regions of DNA to protect it from mutation, or binding to RNA to help regulate how genes are read. But its most surprising role is how it interacts with the misfolded proteins of the brain. The central problem in diseases like Parkinson's is a protein called alpha-synuclein. In a healthy brain, alpha-synuclein aids in neurotransmitter release, but in Parkinson's, it misfolds. Monomers, or single proteins, clump into oligomers, which are small groups, which then lengthen into insoluble amyloid fibrils. These fibrils are the spaghetti I mentioned earlier. They are long, tangled, and toxic to neurons. For years, the scientific dogma was simple. It stated that any clumping of proteins is bad. Early experiments showed that adding spermine to alpha-synuclein in a test tube made it aggregate faster. Therefore, scientists assumed spermine might be a villain, accelerating the disease. However, a pivotal study led by Jinghui Luo at the Paul Scherrer Institute completely flipped this narrative. Using advanced imaging techniques like small-angle X-ray scattering and cryo-electron microscopy, the team watched exactly how spermine interacted with alpha-synuclein. They discovered that spermine drives a process called biomolecular condensation. Instead of allowing the proteins to twist into rigid, toxic fibers, spermine acts as a molecular glue. It pulls the proteins together into soft, liquid-like droplets. Think of a plate of plain spaghetti. The noodles act independently, sliding around and getting tangled in a way that makes them hard to scoop up. Now imagine melting cheese over them. The cheese binds the noodles into a cohesive ball. Without spermine, the proteins form long, sharp fibrils that can pierce cell membranes and kill the cell. With spermine, the proteins condense into a compact droplet. But why is the cheese ball better? The genius of this mechanism lies in the cleanup crew. Our cells have a waste disposal system called autophagy, literally meaning self-eating. Autophagy involves creating a double membrane sac called an autophagosome that swallows trash and delivers it to a lysosome for acid digestion. It turns out that the autophagy machinery struggles to swallow long, rigid fibers. It is like trying to stuff a dry spaghetti noodle into a small trash bag. It pokes through or does not fit. However, the condensates created by spermine are compact and essentially tasty for the autophagosome. They fit perfectly inside. By clumping the proteins into these droplets, spermine effectively packages the toxic waste for rapid removal. In experiments with nematodes, worms treated with spermine did not just have different protein aggregates, they lived longer, moved better, and had healthier mitochondria. The Spermine Defense was proven real. Now, if you search the scientific literature, you might find a confusing contradiction. Some papers claim polyamines are neuroprotective, while others claim they are neurotoxic. How do we resolve this? Biology is rarely black and white. It is a game of Goldilocks. If you have too little spermine, DNA destabilizes and autophagy slows down. If you have just the right amount, spermine induces the protective condensation of proteins and facilitates their clearance. But if you have too much, extremely high levels of polyamines can overstabilize DNA, preventing necessary gene reading or produce toxic byproducts. When spermine is broken down by the body, it produces acrolein and hydrogen peroxide, compounds that cause oxidative stress. The Spermine Defense relies on a delicate balance. In a healthy young brain, spermine levels are tightly regulated, and the defense works. As we age, this regulation falters. There is also a critical challenge involving the blood-brain barrier. While you can eat spermine-rich foods, the blood-brain barrier is notoriously strict. It does not easily let polyamines from the blood pass into the brain. The brain relies heavily on synthesizing its own spermine. This means that simply flooding the blood with spermine might not rescue a brain that has lost the ability to make it. However, circulating spermine does rejuvenate the immune system and improve overall vascular health, which indirectly supports the brain. Furthermore, precursors or specific transport mechanisms under study might allow for therapeutic intervention. To truly appreciate a hero, look at a world without him. We can look at Snyder-Robinson syndrome, a rare genetic disorder caused by a mutation in the SMS gene, the gene that codes for spermine synthase. Boys born with this condition cannot convert spermidine into spermine. The result is heartbreaking and revealing. They suffer from intellectual disability, muscle loss, thinning bones, and seizures. The lack of spermine leads to a collapse of cellular integrity. Lysosomes, the recycling centers, bloat and fail. Mitochondria, the power plants, sputter out. This tragedy underscores that spermine is not just a bonus molecule for longevity. It is an absolute requirement for the development and maintenance of a functioning nervous system. It proves that the Spermine Defense is a foundational pillar of brain health. Beyond the brain, the Spermine Defense operates system-wide. Let's talk about mitochondria. These are the batteries of our cells. As we age, they wear out. Spermine has been shown to enter mitochondria and stabilize their machinery, ensuring efficient energy production. In the study I mentioned earlier, the worms treated with spermine didn't just have cleaner brains, they had younger mitochondria. Then there is DNA protection. Your DNA is under constant attack from radiation and oxidative stress. Spermine wraps around the DNA double helix like a protective sheath. Its positive charge neutralizes the negative repulsion between DNA strands, allowing DNA to pack tightly and shielding it from damage. And we cannot forget the autophagy switch. Spermine and its precursor spermidine mimics the effects of fasting. When you fast, your body runs out of easy energy and switches into repair mode. Spermine tricks the cell into thinking it is fasting, activating the same deep clean protocols without the hunger pangs. This is why it is often called a caloric restriction mimetic. Before you rush to buy spermine supplements, a word of caution is necessary regarding cancer. Spermine is a growth factor. It helps cells divide and protects them from death. In healthy tissue, this promotes regeneration and repair. In cancerous tissue, however, this can promote tumor growth. Cancer cells differ from normal cells in that they have an insatiable appetite for polyamines. They often ramp up their own internal production of spermine to fuel their rapid division. This is why some cancer drugs work by blocking polyamine synthesis. This double-edged sword means that while spermine is excellent for preventing neurodegeneration where cells die too fast, it must be used with caution in the context of cancer where cells do not die enough. This is why dietary sources are generally considered safer than high-dose synthetic supplements, as they provide a physiological boost without flooding the system. So can you eat the defense? While the brain makes its own spermine, maintaining high systemic levels of polyamines, like spermine and spermidine, is linked to longevity and better cognitive health in population studies. The blue zones, areas where people live longest, often feature diets rich in these compounds. The undisputed king of polyamines is wheat germ. Adding wheat germ to smoothies or yogurt is the easiest way to boost intake. The cheese on pasta analogy I used earlier is chemically fitting, because aged cheese is another great source. Fermented aged cheeses like blue cheese, cheddar, or Parmesan are rich in spermidine and spermine because the fermentation bacteria produce it. Soy products are also powerful. Natto, which is fermented soybeans, is a powerhouse, and tofu and miso are also good sources. Mushrooms, particularly shiitake, are surprisingly high in these molecules. For fresh vegetables, green peppers are excellent plant-based sources. Beyond diet, lifestyle factors matter. Intermittent fasting naturally upregulates the body's own polyamine production and autophagy. Gut health is also crucial because much of our circulating polyamines come from our gut bacteria. A diet high in fiber and fermented foods supports the microbes that manufacture these defense molecules for us. The discovery of the Spermine Defense fundamentally shifts our approach to neurodegenerative disease. For too long, we viewed the aggregation of proteins as a simple mechanical failure, a clogging of the pipes. Now we see it as a nuanced biological process, where the type of aggregate matters. We now know that the body has a built-in mechanism, a molecular cheese to handle the spaghetti mess. The failure of this mechanism in aging brains, whether through the loss of spermine synthesis or the overwhelming of the autophagy system, is a key driver of disease. The future of treatment may not lie in synthetic drugs that attack the proteins, but in restoring the ancient, natural defenses of the cell. Whether through gene therapies to correct spermine synthase, drugs that mimic spermine's gluing ability, or lifestyle changes that boost our polyamine levels, we are inching closer to a world where we can help the brain fold its own proteins correctly, preserving the memories and minds that make us who we are. The Spermine Defense is a reminder that sometimes the most powerful medical breakthroughs come from understanding the microscopic custodians that have been keeping us alive for millions of years. Thank you for listening to this deep dive on G Fun Facts Online.